Happy Wednesday. Welcome back. For this review, I'm straying away from that whiteboard because whatever I would try to draw to do for the phases of meiosis, that would look like a train wreck, and I really like the way this video shows it. I think it makes it a lot easier, so no worries. You don't have to look at my drawings today. Okay, so what did we talk about yesterday? What even happened? For meiosis, remember there are two phases of meiosis to make your 4 haploid sex cells. One point that I really want to hit on that I don't think I made super clear yesterday. Before any type of cellular division, whether it's mitosis or meiosis, before anything like that ever happens, your DNA has to be replicated. It has to have a copy of it made. So when your cells actually split off, they have the proper number. So before meiosis happens, during interphase, your chromosomes are going to be copied. They're going to be replicated. Then through meiosis 1, that cell is going to form 2 new cells that are going to be genetically different, and then those 2 new cells are also going to split into 2 during meiosis 2 to make your 4 haploid cells, and see how they are different. Those chromosomes don't look identical. Okay, so what actually happens? So again, during interphase, see how those chromosomes are duplicated. Now they are ready to go through that cellular division. Meiosis 1 is going to start eventually, here we go, with prophase 1, because remember PMAT, during prophase 1 those chromosomes condense, that nucleus breaks down, and then the very, very important step of meiosis is going to happen, that crossing over. So they literally are going to go on top of each other, those homologous chromosomes, and they are going to swap some of their genes. You may see that called genetic recombination. It's a new combination of genes. So they are no longer looking how they used to look. They made some trades. So then during metaphase 1, M for middle, they're going to line up side by side right in the middle. And then eventually anaphase 1 is going to happen today. Hurry up. There we go. Anaphase 1 is going to happen, and then they're going to move away from each other, and then we'll see telophase 1, where those 2 new nuclei are going to form around the now separated homologous chromosomes. Okay, so then we have cytokinesis happening, where it snips those 2 cells apart, so we have 2 new cells. Okay, so that is the end of meiosis 1. Then we hit up meiosis 2. And remember, for meiosis 2, we do not replicate or duplicate or make a copy, however you want to say it, those chromosomes. Whatever chromosomes are in those 2 new cells after meiosis 1, that's what you got. Okay, so then we go through all of PMAT once again. You are going to see, this thing is not moving at the pace I want it to move. There we go. You're going to see prophase 2, where the nucleus breaks down again. But remember, no crossing over happens. That only happens in prophase 1. So eventually they're going to line up in the middle, just like that. And then during anaphase 2, the chromatids, or those 2 lines in that X, they are now going to separate. So you can see, and I'm actually going to pause it because this is gorgeous. This is exactly what you need to see. So now that those X's, or those chromosomes, have separated, you can see that the genes on both of those chromatids are not identical. They are different, and whichever side gets whatever genes is totally random. It's not really too predictable. So in this cell we have, look at all that green, that's like 1 gene, and then we have the blue, and then over here it's all green, so they're not identical. Okay, telophase 2. You're going to have those new nuclei form around the chromatids. And then cytokinesis, snip, 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 snip. You have 4 haploid cells that are genetically different. And they're haploid, meaning half, because you didn't have that second round of replication happening before meiosis 2. So they have half the amount that the normal cells would normally receive. Okay. So let's look at our Guided practice. Here is just another layout of what happens in meiosis 1 and 2, so you can see it differently. The more you see it, the easier it's going to get. So we have two little questions here that we just want to answer together. How does meiosis 1 differ from meiosis 2? So what is different about both rounds of division? Well, first, in meiosis 1, we can say, let's put number 1, meiosis 1 has crossing over. So those chromosomes in prophase 1 are going to cross over. Also, and it actually says it right there in the picture, the homologous chromosomes are separated in meiosis 1. Okay, so then what's different from meiosis 2? So in meiosis 2 has no crossing over. Okay, those chromosomes do not swap their genes anymore. And then the sister chromatids, which again, those are the two lines that make up the X, those sister chromatids are separated. Okay, so what causes, and this is super important, what causes the daughter cells or the brand new cells made during sexual reproduction to be genetically different? They are different because of crossing over, or we're also going to put genetic recombination of the homologous chromosomes in prophase 1. Okay, in mitosis, nothing gets swapped around. They just make direct copies, and that's asexual reproduction, because remember, asexual makes identical. This is sexual reproduction, and in meiosis, the chromosomes get swapped around. So they are genetically different. All right, so what do I need you to do today? If you scroll down, let me explain this nice and well. Let me make it clear for you. In the show what you know, you only have to do one of the following. You're either going to do choice 1 or 2. You do not have to do both. If you want to do both, by all means, go ahead. But I'm only going to really count one of them. Okay, so choice 1. On a piece of paper, you can draw out each phase of meiosis 1 and 2. So give me prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2. You get it. Draw all of that out and make sure you include the chromosomes and what is happening to the chromosomes and label each phase. Now, since you're drawing it out, you do not have to explain or write down what's happening, because I should be able to see that in your picture. So I actually attached an example of a drawing just so you can see what I'm talking about. What am I even looking for? If it ever loads, right here. So make sure you draw those chromosomes. You probably want to use different colors so you can actually see those genes get swapped, and just label what phase is happening where. Okay, if drawing isn't your thing and you're like, Miss Long, for the love of God, don't make me do that, your other option is to create a quizzes. Not do a quizzes, you're going to create your own. So, and I label exactly what I want you to have in your quizzes right here, but let me show you. If you go to quizzes, create a quiz, after you log in, of course, it is going to ask you to, the Wi-Fi is so slow today, ask you to name the quiz. Let's name it Meiosis. That makes sense. And this is science. Next. Okay. It's going to ask you to put in your own questions. So for each question, I want you to have one question for each phase of meiosis. Meiosis. Sometimes I'll say meiosis on accident, and I'm sorry for that. I need to say meiosis. Okay, so for each question, I want a phase of meiosis. So like this one can be prophase 1. Now the answer choices, it's a quiz, so one choice should be correct. Describe what is happening during that phase. You can say the homologous chromosomes cross over and nucleus breaks down. And make sure you mark that as the correct answer. And then, of course, like any quiz, have some wrong answers. So you can say chromosomes line up in the middle. That doesn't happen in prophase 1, that's metaphase 1. So don't mark that as correct. So go through, save each question, and then when you're done, you should make sure it's public. It will ask you to share a link, so just copy and paste the link for this quizzes and drop it in this box here. And if you do the drawing, take a picture of it, upload the picture in that box there. Whichever one you do, put it right here. Okay, once you're done with that, you have this table to fill out, just comparing meiosis and mitosis. And then your exit ticket is going to be on a Google form from now on. So just go back to the assignment, open up the exit ticket, answer the question, and justify your answer, meaning explain how you knew that was the correct answer. Okay, and as always, there is extra stuff down here to help you in case you are totally lost. And of course, I'm always here to help you. Some of you are just putting, I don't know, or you're confused. Hit me up, that's why I'm here. I'm literally sitting in this classroom all day just waiting for you to email me. Okay, good luck. Let me know if you need anything.